No matter where I've lived, I've always been drawn to coastal waterways. I think it's in my blood. I grew up on the Oregon coast, and I got to spend a lot of time in these areas as a kid. When I started graduate school at Portland State University, it seemed like a natural fit for me to return to these habitats. My name is Sarah Kidd, and I have spent the last 8 years researching wetland restoration in the Columbia River estuary. Throughout the world, wetlands have been diked and drained to create more room for agriculture and development. As we have learned about the impacts that these practices have had on our natural resources, we have developed strategies to recover some of the lost habitats and the ecosystem services they provide. Over the last decade, restoration work has been in full swing to convert agricultural lands back into tidal wetland habitats. Essentially, this is done by removing the levee barriers that were put in place to prevent these areas from flooding, and therefore reintroducing them to the tidal flow of the estuary. Here in the Pacific Northwest, thousands of acres of restoration work has taken place. My research has been focused on tidal wetland habitat recovery in Youngs Bay, which is a tidal wetland complex located at the mouth of the Columbia River estuary. I've spent many hours surveying the plant community and soil development of these newly flooded landscapes in an attempt to track changes in their ecology over time. It's important for us to understand how and if these habitats recover once restoration actions have taken place. Restoration work also provides us an opportunity to ask questions about how climate change and sea level rise may affect these habitats in the future. Restoring flooding provides a proxy for evaluating the impacts of sea level rise, which is expected to increase flooding and salinity levels throughout the estuaries of the world. Through my research, I have discovered that many restored wetlands in this region suffer from non-native plant invasion from species such as reed-canary grass and common rush. These non-native agricultural holdouts reduce the habitat quality of these wetlands for salmon and other wildlife. Working with the Lewis and Clark National Historical Park, I developed a study to take a closer look at the mechanisms driving these non-native plant invasions and to evaluate the potential impacts of sea level rise on these habitats. We chose to focus this work on the seed banks of these wetlands. The seed bank is essentially all the viable plant seeds found in the soil. The seed bank is important because it provides a template for wetland plant community growth and resilience over time, and this is especially critical after restoration when wetland plants have to reestablish themselves in these newly created habitats. After extracting seed bank samples from the dominant native and non-native plant communities in the National Park's restored wetlands, I brought them back to Portland State University, and then with the help of my interns, I processed them in our soils lab. Processing entailed removing the excess organic matter and roots from the seed bank samples and then getting them ready for grout in the greenhouse. The action happens. We are in the greenhouse. This is greenhouse 2, and this is where I have the seed bank study all set up. We're in the greenhouse, and what you see in here is all the species that have grown out of the seed bank samples. Once they mature and germinate, we transplant them and grow them to maturity so we can identify them. And so what you're seeing here, these are all seeds that germinated out of the seed bank, and then we pulled them to grow, and we just had thousands of seedlings germinate in the greenhouse. It's been amazing. So this is where the action happens. These tubs are all of the different flooding and salinity treatments. So we have seed bank samples collected both from the native and the non-native plant communities within the national park wetlands. And then those soil samples have been distributed amongst all of these pots and all of these treatment tubs. And they're all color-coded and numbered, and we know where all of them are. And so all the seed bank samples samples are treated to 3 different flooding treatments and 3 different salinities, and those are also color-coded in these tubs. And then under the tub, if you come down here with me, these are the water reservoirs which hold all of the treatment water. So we are able to control the salinity really specifically by having each salinity treatment have its own water tub. So we check this twice a week and we correct it. So at high tide, all of these fill with water, and then there's this drain here, you can maybe see it, which keeps the water level below the top of the pots. And that just allows all these pots to stay independent samples so that the water's not overtopping and distributing the seeds everywhere. They're all staying very wet at high tide, but they're also not distributing amongst each other. In previous work, I had found that in newly restored sites, the non-native species tended to dominate the high marsh regions, while the natives were more successful in the low marsh. By examining the seed banks and treating them with different flooding and salinity levels, I was attempting to identify the mechanisms that might be driving the high to low marsh zonation of these species observed in the field. For over 5 months, we went through and identified and counted seeds and seedlings by hand. By the end of the study, we had counted and identified over 23,000 seedlings from 43 different species. Results from the greenhouse study confirmed our suspicions. The non-native species were prolific seed producers. Non-native seedlings vastly outnumbering those of common native wetland species, such as carex and bulrush. Non-native species germination, however, was suppressed by a combination of frequent flooding and increased salinity conditions, characteristic of low marsh wetland zones. We also found that native species germination was similar across all the flooding and salinity treatments, indicating that these species are better adapted to withstanding the stress imposed by more frequent flooding and higher salinity conditions. These results indicate that issues with non-native plant invasions, especially reed-canary grass and common rush, are likely tied to their overwhelming presence in the seed bank and their ability to germinate readily and likely outcompete the native species under existing restored high marsh conditions. These findings are important as they help us have a deeper understanding of the ecology behind these plant invasions and help us develop strategies for managing them in the future. Locally, sea level rise may result in pushing these non-native species up the elevation gradient. However, we may lose important high marsh habitats in the process. As we move forward and continue to conduct these large-scale restoration projects, we'll need to start to pay extra close attention to the soil under our feet and the environmental conditions we create in the process.